Good morning, everyone. I'm the moderator for today's webinar, so please wait for a few minutes. We will start the session soon. Thank you very much. Hello everyone and hello from Malaysia. Welcome to the IMS Biotech and Bioinformatics Student Association's APSA webinar series 2022. So you have joined our webinar on phage therapy and alternative therapy to combat antimicrobial resistance organized by IMS Year 3 Biotech and Bioinfo students. So thank you for taking time out and being here today. And we will begin our program shortly. So I would like to kick off today's program by reminding you some of the housekeeping items as seen on the screen. So first of all, please turn off your microphone during these sessions and only unmute when you are required to speak. Please turn on your camera so that we can see each other. So if you have any questions or comments, please feel free to message us on the chat box. We will try our best to address your concerns. So the chat box is also there for you or to introduce yourself. Please give us your name and your organization. Last but not least, no phone calls or disruptions during this session. Note that this session will be recorded and live on YouTube. The recording will be saved on the YouTube channel, AIMS webinar, right after this session. An evaluation survey will appear at the end of our session. Please spend some of the time providing your feedback as it will be really meaningful for us to improve our next session. So thank you very much for your attention and once again, welcome all to our webinar. My name is Chai Wei Chi and I'm proud to tell you that I'm a BSc Biotechnology Year 3 student at AIMS University. So it is my honor to serve as the moderator for today's webinar. So if anyone has any issues with audio or video, please use the chat box and we will do our best to help you. To start the session, I would like to invite the Dean of the Faculty of Applied Science of AIMS University, Senior Professor Manikam Ravichandran, to say a few words, please.
Okay, good morning, everyone, on a Saturday morning, and thank you for gathering for a nice topic on phage therapy. First of all, I'd like to thank Chai Ho Chi thank you for introducing, and all the Ms. Vinodini and all the APSA member, everyone. and Vivian and all. So you have been consistently organizing. So my hearty congratulations for the job well done by AIMS Biotechnology and Bioinformatics Student Association, APSA. You have been rocking and you are doing a wonderful job of doing a cutting edge topic, promoting biotechnology and STEM in Malaysia, I think consistently for the last few years. At this outset, I would like to welcome Dr. Adeline Song Ai-Lian, and thank you so much for your time and on a Saturday morning and sparing some time with the students, sharing your knowledge on a relevant topic. I think if at all they're talking about next pandemic, the antibiotic resistance is going to be the next one, because we are losing choices of antibiotic and there is not many new antibiotics coming in. So we have the weapon, the one of the largest number of living organism on this planet of Phage. Every there's more than 10 to the 31 number of phages in this world. Every bacteria has a predator phages out there. So we have a treatment in the soil, in the sewage water, everywhere it is there. How we use it, how we utilize it effectively, that's more important. So I think Dr. Adeline, thank you so much for your time all the way from UPM. So we really appreciate your sharing of knowledge and hope the audience are interactive. And so basically we are mostly biotech students and some of the faculty members from AIMS University all are here. I think we also have some international participants. So it's a beautiful campus. If you have a chance, you come and visit us. Take it as a personal invitation. So you can come, and now the borders are open from 1st of April. I think everyone follows the SOP and stay safe. So it's a nice campus to come and visit, and it is located in a nice green campus, and it is located at the Bujang Valley in Kedah State in Malaysia. So thank you so much, Jose, and thank you for the opportunity to talk to you. And I don't want to give a long lecture here, so I'll pass it over to the MC. Thank you.
So thank you, Prof. Ravi, for your welcoming speech. Now I would like to invite the chairperson of the webinar, Vinodini Kunalan, to say a few words. Vinodini, the floor is yours.
Hello everyone, and hello from Malaysia. On behalf of AIMS Biotechnology and Bioinformatics Student Association, APSA, I welcome you all. I'm Vinodini Kunalan, a final year student in BRC Honors Biotechnology degree program of AIMS University. It is an honor to serve as chairperson of the organizing committee. As a chairperson, I would like to express my sincere appreciation to our committed team who worked hard to organize this webinar. Thank you, Dr. Adeline Song Ai-Lian, for accepting our invitation. And thanks to the Dean of our faculty, Senior Professor Manikam Ravichandran, for the full support. And I would like to thank all the participants from the various countries for joining us this webinar. I welcome our honorable speaker of the day, Dr. Adeline Song Ai-Lian, Senior Lecturer from Department of Microbiology, Faculty of Biology and Biomolecular Sciences, University Putra Malaysia, UPM. Senior Professor Manikam Ravichandran, Dean of our Faculty of Applied Sciences, AIMS University, and our very supportive professors, lecturers, ladies and gentlemen. I'm delighted to see all of you here. Finally, I would like to thank the organizing committee for doing an excellent job. I hope you will enjoy and gain some insights from today's webinar. Once again, thank you. Ms. Chayuchi, over to you.
Thank you, Vino. So, as we all know nowadays, antimicrobial resistance pose a major threat to human health around the world. So, and infections due to antimicrobial resistance cause millions of deaths each year. However, phage therapy is discovered to be a significant alternative to antibiotic in the current era of multi-drug resistant bacterial infections, and the success rates found is in this study were 80 to 95%. So now it is my great pleasure to introduce Dr. Adeline Song Ai-Lian, who give us a detailed look and discussion on this topic. So currently, Dr. Song is working as senior lecturer at the Department of Microbiology, Faculty of Biotechnology and Biomolecular Sciences, University Putra Malaysia, UPM. So Dr. Adeline Song's primary research interest is in lactic acid bacteria and phage therapy. She has published over 32 research papers in national and international peer-reviewed journals. She is also an EXCO member of the Malaysian Society for Microbiology and the Malaysian Society for Lactic Acid Bacteria. Her current research is on the use of lactic acid bacteria as a delivery system for therapeutics and phage therapy endolysin as an alternative antimicrobial resources. So with this little background, I would like to request Dr. Adeline Song to take the floor charge and start the presentation. Dr. Adeline Song, the floor is yours, please.
All right. Can you hear me?
Yes.
Yes.
Okay. All right. That's great. Thank you very much. Good morning, everyone. Good morning, Prof. Ravi Chandran, the Dean of the Faculty of Applied Science, AIMS University, the chairperson, Ms. Vinotini, the moderator, Ms. Chai, as well as everyone present here. Thank you very much for inviting me to give a talk on phage therapy. Let me just try to share my slides first. And okay. Can you see that?
Yes, yes, we can see it.
Okay, so all right, so the topic that was given to me today was phage therapy and alternative therapy to combat antimicrobial resistance, or AMR. I have to admit that I wouldn't call myself an expert in phage therapy. I've only been researching this field for about 4 to 5 years. So I think there are many people, even in AIMS. I know that AIMS is very active in doing research on phage therapy. So probably your lecturers have much better knowledge than I. So this is really more of a sharing session. So I will be sharing a bit about just very general about AMR and phage therapy, and then I'll also would like to share with all of you a bit of what we are doing, or at least my research group is doing in UPM, and hopefully we can actually exchange ideas and collaborate in future. So as I was saying, I have only been researching phage therapy for the past 4 to 5 years or so. Before that, I was actually working more on lactic acid bacteria. I do genetic engineering in lactic acid bacteria and use it for as a delivery system and a microbial cell factory. So currently, I'm also trying to merge these two research areas together, phage therapy as well as lactic acid bacteria. And so I will actually talk a bit more about it later on. All right, so without further ado, let's go to the first slide. Okay, so what's antimicrobial resistance? I think all of you probably know this. So there is a lot of definition in, okay, before that, let me just see what time it is so I don't go overboard. So I would appreciate it if the moderator could, if it's like too long already, you can let me know, that I will, quickly wrap it up.
Yeah, Dr. Adeline, for sure. I will remind. Okay, thank you.
So antimicrobial resistance, there are many definitions, but WHO defines it as a, it occurs when bacteria, viruses, fungi, parasites change over time. And the key word here is that they are no longer responsive to medicines. Okay, and it makes infections harder to treat. And that's why it increases the risk of disease spread, severe illness, and death. So when we talk about AMR, there are actually many types of AMR. There is antibacterial resistance, antiviral resistance, antiparasitic resistance, antifungal resistance. But today we'll only be touching about antibacterial resistance. And when we talk about antibacterial resistance, we usually talk about antibiotics. All right. And the reason why is because even though AMR is a broad term, but currently the major concern is with the resistance of bacteria to antibiotics. And also because we are talking about phage therapy today, and phage therapy only addresses this concern. All right, it's only an alternative to antibacterial resistance. It doesn't affect the other types of resistance. All right. So antibiotics, as you all know, I think this is a very famous picture for microbiology and biotechnology students or life sciences students. This is Alexander Fleming. and he's the person who discovered penicillin in 1928. So antibiotics is very, very effective and very easy to use. And since then, it has been our first line of defense against bacterial infection. All right, all this while from then until now. However, there is a problem with antibiotics, using antibiotics. Oops, sorry. And this was actually how to say, it was actually already foreseen by Alexander Fleming himself. So he said, the time may come when penicillin can be bought by anyone in the shops, then there is the danger that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant. So this was actually said by Alexander Fleming in his Nobel lecture in 1945. So antimicrobial resistance has been, we have actually foreseen it a long time ago. It's not exactly something new. Okay, so antimicrobial resistance is a global problem. If you look at this chart, you can see that the date of discovery from the first antibiotic, which is penicillin in 1928. And then over the years, there has been a few classes of antibiotics which have been discovered. But the problem is that after every year, a few years after it has been discovered, you also get resistance identified. All right, so in the case of penicillin, it's like 1928 and 1940, there's a resistance has been identified. And then for some, for example, tetracyclines, it's very fast. Right after you discover it, a few years later, there's resistance. All right, some of them take a bit longer, like the macrolides, but every time a new class of antibiotics is found, very soon later, resistance is also identified. Also, there's been almost 30 years since a new class of antibiotics was last introduced. So we have new antibiotics, but the antibiotics are usually derivatives of the current antibiotics. All right, so a totally new class of antibiotics is very hard to find a new class of antibiotics, and it's also difficult to discover antibiotics which are able to overcome antimicrobial resistance at this point. All right. So causes of AMR. So I took this from the American Society for Microbiology, development of antimicrobial resistance. So how it happens here, it shows that antimicrobial products are used to kill or significantly slow the growth of disease-causing microbes. So you have a population of microbes, but the problem is that sometimes when you use antimicrobial products, and they are not at the lethal dosage, they are sublethal dose. And what happens is that not all the microbes or the bacteria dies. So you have some which survive. And under selective pressure, these microbes, they are able to evolve, and they have ways to actually overcome the antibiotic. So, and these few very strong bacteria will grow to a larger population, and they themselves can actually transfer their antimicrobial genes through horizontal gene transfer to a new group of bacteria. So this is how AMR can actually spread. So the key causes of AMR, there are a few that are identified here. So the ones on these are the ones that are commonly known. So for example, overprescription of antimicrobials. We know, like I mentioned just now, antibiotics is actually our first line of defense against bacterial infection. But a lot of times, when you go and see the doctor, sometimes if you have a cough or a cold, they don't even really identify whether it's a viral infection, a bacterial infection. They will just give antibiotics for prevention, right? And you'll also know that when you go see a doctor, the doctor will always tell you to make sure you complete your course of antibiotics. Not everybody does it. A lot of times you stop your course of antibiotics because you feel that I'm fine, I'm better already, so I don't need to complete. And this is why this is also called subdosage of subdose of your antibiotics. So you don't really kill all the microbes that you're supposed to kill, and you will have, subpopulations of microbes which are resistant. So this is another reason why it's important for you to finish your the whole course of antibiotics. But other than human consumption of antibiotics, a very, very important or very, very a contributing factor of AMR is actually the use of antimicrobial overuse in livestock and fish farming, in agriculture and aquaculture. So the problem here is that a lot of farmers, they actually use antibiotics as not just to treat infections in their cattle, in their animals, but as a prevention. So before the animals even fall sick, they are already giving them antibiotics so that their animals, to prevent the animals from falling sick, so that they don't have a loss, in economy and to lose their livelihood and everything. So this is a big problem, because that's why they are called growth factor, what AGP, antimicrobial growth promoting factors or something. Yeah, so they actually are trying to give the antibiotics to sort of promote the growth so that they don't fall sick and they can actually grow well. And this is a huge problem, because imagine how much of chickens and cattle that we are eating every day, and that's the amount of antimicrobials which are actually circulating in our environment every day. All right. So it doesn't matter whether is it human or is it animals. The point is we are all connected in life, and so even if it's in animal, the animal also will, they have their feces. The feces go into the soil. Even if it's on meat, the meat also can be eaten by humans. The bacteria, if it's not properly cooked, it will also go into humans. If you have a person who is sick and is having a bacteria which is resistant, he can also pass it to someone who is not resistant through horizontal gene transfer. So it keeps on circulating in our environment. All right, so this antimicrobial resistance has become a big issue, a global problem. So this is a chart to show the global AMR burden. This is from a review paper in 2019, and it shows that the death of that is associated with antimicrobial resistance is about, the rate is about 100 in a population of 100,000. And this is according, the different colors are actually according to different regions of countries. And you can see South Asia is pretty high. It's in fact the highest after the sub-Saharan Africa region. And yeah, so it's a concern also in where we live as well. So this is talking about the rates, about 100 in 100,000. But then if you look at this paper as well as some news articles, you will see that the amount is about 5 million deaths in 2019. So it says here that the third leading global cause of death is likely not what you think. So we always think that it's things like heart attack or cancer, but now the third leading global cause, according to this article, is actually associated with antimicrobial resistance. All right, and then you have also headlines like this: superbug resistant to 2 last resort antibiotics found in US for the first time. So while we have been using antibiotics for many years now, until we have come to the certain antibiotics, like for example colistin and carbapenem, which are actually called the our last defense of antibiotics against bacterial infections. And even then, we already have superbugs which are resistant to them. So where else do we turn to? Because these are supposedly our last resort antibiotics already. All right, and then another one, superbugs to kill more than cancer by 2050. This is a prediction done by a group of experts, and they said that in 2050, the amount of antimicrobial resistant associated deaths will be even more than cancer. So these are all very scary headlines, all right? So now do you know what the problem is, antimicrobial resistance? So there is this race to actually, find alternatives to antibiotics. So there are many types of strategies being used by researchers. For example, and I'm sure that you have heard many of these options as well. So you have like antimicrobial peptides, for example, bacteriocins. Some think vaccines is the way to go. Instead of killing the bacteria, you sort of make yourself immune to the bacterial infection. And you have a crystal-Cas9 system, you have probiotics. So a lot of like farmers, they are actually, because now that we are trying to limit antibiotic usage, so a lot of industries the government is actually clamping down on these industries. They are not allowed to use antibiotics. So instead of using antibiotics as growth promoting factors, they're actually using things like probiotics to help the animals actually grow well and not fall sick. Okay, and then we have like things like plant extracts, essential oils are also something which are being researched quite extensively. You have quarantine symptom inhibitors such as biofilms and things like that. You have antibodies, and of course you have fish therapy. So today we'll be talking about fish therapy. All right. So what are bacteriophage? Phages, or they are short for bacteriophage. They are viruses which infect bacteria. I think all of you know this. It was actually first discovered by William Twat in 1915, but in 1917, the term bacteriophage was coined by Felix de Herel, which basically means bacteria eater. So it's very nice. This system is very nice because phages are actually natural killers of bacteria. And they are also very abundant, like what Prof. Ravi mentioned just now. They are the most abundant organism in the world. All right. So there are two types of phages, lytic or temperate, also known as lysogenic phages. And this is also very general. I think you all know that. But actually, there are more than two types of phages. There's also like pseudo-lysogenic phages. There's also phages who go through chronic infections, life cycle, but these are not very well studied. So the most well studied cell cycles is actually the lytic or temperate or lysogenic phage. All right, so looking at the life cycle of phages, this is the lytic life cycle, where the phage will actually infect a host cell. Then it actually injects the viral DNA, the phage DNA replicates, and phage proteins are made. And then new progenies are found, and then the phage will lyse the cell breaks and release new phage. Okay, so this is the lytic life cycle. And then the lysogenic life cycle, where the phage actually infect and then injects the viral DNA. However, the viral DNA incorporates into the host genome. And when the cell divides, the viral DNA also is passed to the daughter cells. And then when the conditions are non-conducive or something, then they will enter into the lytic phage. So it makes new progenies, just like the lytic phages, and then it's lyse. So the lysogenic life cycle, the lysogenic phages will also go through the lytic life cycle. All right, but the lytic phages do not go through the lysogenic life cycle. All right. So is phage therapy new? All right. So why is it that we keep on hearing about phage therapy now? But phage therapy has actually been used for a very, very long time. So for example, in 1919, there was a first documented clinical use at the hospital. This is a French hospital. I don't know how to pronounce it. All right. And in this first documented clinical trial, phage was successfully used to treat 4 pediatric cases of bacterial dysentery. And then in 1931, there was a trial in Punjab for cholera, India, involving 118 control subjects and 73 experimental subjects, where 90% mortality reduction was reported. So there have been many cases like this throughout the years, but the major challenge was that there was inconsistent reporting. Some of them said that it worked very well. Some people said that, oh no, it's like there's no difference between those that are treated with phages and those that are not. And this is, at that time, is a lot of the, when it didn't work, a lot of it was due to poor knowledge on phage biology at that time. They didn't know like at what conditions the phage was stable. They didn't know like what was the required titer or the best ways to actually administer the phages. So there were a lot of unknowns then. And with the advent when antibiotics was found, most of the Western world, America and all, they actually stopped with phage therapy. They put it aside because antibiotics were working so well. All right, so there was no, no need for phage at that moment. All right, so only the USSR, Russia, and Eastern Europe continued their research on phage therapy, and this is a institute which specializes in phage therapy in Georgia. It's a European country, all right, and it has been around from the 1916 till today. So it has a very big library of phages that can be used, all right. So if phage therapy is not new, why the new interest in phage therapy? All right, it has been around for almost hundreds of years, 100 years, right? So what's with the new interest in phage therapy? So the problem now is that we have this antimicrobial resistance. It's becoming more and more serious, getting out of hand. So that's why phage therapy is being revisited again. And also with the current information and the current technology that we have, we could actually, revisit phage therapy, and it might be a very good potential. It has a good potential as an alternative to antibiotics. All right. Okay. So pros and cons of phage therapy. So we look at the pros first. On one hand, they are bactericidal in the sense that they really kill off bacteria. Some antimicrobial agents, they are bacteriostatic. They don't kill the bacteria, they just slow them down. So phages are bactericidal, so that's good. They have a very specific target host range. So if you look at the pros and the cons, they are actually, this point is actually in both the pros as well as the cons. The reason why you want it to be specific, because like certain things like antibiotics, they are very broad spectrum. They kill the bad bacteria as well as your good bacteria. So you know that you have a lot of good bacteria, especially those probiotic lactic acid bacteria in our bodies, who make up our microbiome and actually give us a lot of good benefits that actually improve our health. So when we take antibiotics, we kill that off as well. So we don't want that to happen. We want it to be specific to kill only the bad bacteria. But at the same time, sometimes phage can be so specific that they can only kill very specific strains of bacteria, and that's not good, because if you wanted to use it for a clinical use, if let's say you're being infected by a bacteria strain and your phage is too specific, it may not even be able to kill that particular strain that is causing the infection. So that's not a good thing. So you want it to be specific enough that it doesn't kill the good bacteria, but not so specific that you can only kill 1 or 2 different strains, and then that's it. Then it will not be of clinical use. All right. Another good thing is that it's easily isolated from the environment. As we mentioned just now, it's the most abundant organism, so which means that the likelihood or the chances of finding a bacteriophage that works against a bacterial infection is much more higher than finding a new antibiotic. All right, it has auto-dosing property, which means that you only need to put, theoretically, you only need to apply it once, and then the bacteria, when it's infected by the phage makes more phages, and if you have more bacteria, you also have more phages, right? So the phage will sort of counter, compared to using antibiotics, you have to keep on dosing, increasing the dose, and keep on applying new dosage. All right, so this is what auto-dosing means. You have a narrower potential of inducing resistance. So compared to antibiotics, resistance also happens in phages, but because it's a live microorganism, it can sort of, counter the resistance as well. So the bacteria can gain resistance to the phage can also gain, try to resist the bacterial and try to modify so that it can continue infecting the phage. All right, some phages have biofilm clearance. And then there's also the formulation versatility. You can, if your phage is too specific, you might be able to use something called a phage cocktail, where you use multiple types of phages together to actually increase your efficacy. All right, and low environmental impact. All right, because it's naturally found in the environment and anyway. So the cons, like I mentioned, specific target host range, toxicity concerns. Although phages very specifically only live bacterial cells, they only target bacterial cells, there are still toxicity concerns because the phage may carry genes which are toxic to humans, and especially if you are the phage is a lysogenic phage, it can incorporate into the genome and be transferred as well. Okay, and then not all phages make good therapeutics. So although it's very easy to find phages, but not all of them are good therapeutics. Okay, and I will talk about this a bit later. Okay, and lastly, finally, of course, it can still induce resistance. So we also have, in fact, it's actually quite common that when you use phage therapy, after a while, you can also identify resistance as well. All right. So it's just a typical workflow of how we actually isolate phages. So you have a choice of isolation host. So it depends on what phage you want to isolate. So let's say you want to isolate against E. coli, then you have to have the host first, because the virus only propagates in the host. All right. Then you collect your environmental sample. It may be from soil, from water, from wherever. Usually you will find the phage where the host is found. All right. And then you do enrichment. Then you do detection of plaques, propagation and purification, and then you characterize the host range and other properties. All right. So how to detect? And usually we'll just do a spot test to screen. So you want to look for clear zones like this, right, and not turbid zones. And then you can also do something called a double layer agar, where you do serial dilution, and then you plate it on a top layer of agar, a soft top layer of agar on another layer of agar, and the plaques will form like this. And this way you can also calculate how much of PFU, plaque forming unit, so that you know how much of phages is present. So when we do purification, we tend to take from single colonies like this, single plaques, sorry, and then we will isolate it and subculture a few times until we get individual pure phages. All right, so characterization. The common types of characterization is a one-step growth curve like this, because we want to see things like the burst size, like when the phage bursts, how many of when the bacteria bursts, how much of phages that it releases, how long is the latent period, so on and so forth. And then we do something like efficiency of plating. This is to see whether it can infect what many types of hosts, and whether each host how well it infects compared to the original host. All right, and then you have something like you can also do TEM to get phage morphology like this, so you know what kind of phages it is. And then whole genome sequencing. Now whole genome sequencing is pretty cheap, so most of the time when you do characterization of phage, you also have to send for whole genome sequencing. All right, so what are the desirable characteristics for phages that can be used for phage therapy? You need to have a host range which is broad enough to target several species, but not so broad that it targets good bacteria, like I mentioned. It has to be obligately lytic. All right, we don't want a lysogenic phage because, as I mentioned just now, a lysogenic phage can incorporate into the genome of the bacteria, and that's not wanted. So a lytic phage is the way to go. But a lot of times, in fact, a lot of the phages that we have are actually lysogenic phages. So nowadays, we also, like I mentioned just now, we have new technologies. We can actually also genetic engineer the lysogenic phage and make them lytic, all right, by removing things like their integrase genes, certain genes that are involved in the lysogenic life cycle. You can actually use genetic engineering to change the life cycle, all right. You have to make sure that there are no toxin genes inside. So that's why this is whole genome sequencing is important, to make sure that there are no toxin genes present. And the phage virulence must be good enough that it can actually kill the bacteria fast, and so that the bacteria doesn't have time for it to gain resistance. All right. So approaches to in phage therapy. What I mentioned just now was the conventional phage therapy, where you just directly administer. You have a phage, you isolate it, you characterize it, then you directly administer it, the natural lytic phages, to the bacterial infection. So this is the conventional phage therapy. Then you also have modified phages. There are many genetically engineered phages nowadays for customized purpose. I will talk more about this later on. And then you have phage-derived proteins, where the application of phage proteins with antimicrobial properties, usually this is endolysins. I will also talk about this because this is actually my main research area. And then you have combinatorial therapy, which is to use phage with some other antimicrobial. For example, you can use phages and antibiotics at the same time. So it doesn't have to be like phage will replace antibiotics. They can also work hand in hand, complement each other, so that you don't have to use as much antibiotics, all right? Or antibiotics which are previously not working, with combination of phage therapy, you can make them work again, all right? So this is combinatorial therapy. So talking about advances in phage therapy, like I mentioned just now, we have a lot of genetic engineering tools nowadays. So there has been a lot of work that has been done to engineer the phages to increase things like enhanced lethality. So for example, a phage can express a biofilm dispersing gene, so that it actually helps to eliminate the eliminate the bacteria easily. You can also modify the phage tail to recognize a broader host range. So if, for example, if your phage is too specific, you might want to modify the phage tail so that you can recognize more different strains of phages. And then, like I mentioned just now, you can engineer temperate phages so that it can become lytic phages. You can engineer stable phages. For example, this is a phage. They actually engineer this phage to display phospholipids on the phage, so that the phage can actually survive the gastrointestinal tract. So this is for oral delivery of phages, right? You don't want it to be broken down in your stomach and your gut, so you can actually have additional protection, okay? Something like encapsulation, but this is actually being produced by the phage itself. All right, and then you can engineer phage as a delivery vector. For example, you can deliver genes to resensitize bacteria to antibiotics. You can also deliver CRISPR-Cas9 systems to disrupt bacterial virulence. So for example, the phages can actually be engineered to actually deliver the Cas9 CRISPR-Cas9 system, so that in the bacteria, it will actually turn on the bacteria's CRISPR-Cas9 system to actually disrupt bacterial virulence. All right. And also, these are engineering of the phage itself, but also you can engineer the phage protein. All right. So this is just if you're interested, you can actually look at this paper. This is a review paper on engineered bacteriophages. All right. So success stories. This is a very famous story because it happened to two professors in the University of California, San Diego School of Medicine. where this couple, both of them professors, they went on holiday, and the husband actually got infected by a bacteria, which it was resistant, antimicrobial resistant bacteria. I think it was the Acinetobacter baumannii strain. And so they tried many things but couldn't work, and he was dying. So his wife actually suggested him to use phage therapy because she learned about it during her undergraduate years. Okay, so she went and do some research, and they actually managed to find contacted some people, and then FDA gave approval for them to do, because everything else was failing. So they actually managed to find phages that was able to cure his bacterial infection. So he's alive and healthy now, and they are very well advocating for phage therapy. There's actually a documentary about them. You can go and look up. I think if you just put UC San Diego Health phage therapy, it will come out. Okay. So there's many other stories like this, clinical success. This is just one slide of it, but there's more than this, which you can actually go and look up. All right. So so that's the overall or the big picture about phage therapy. So the next part of my presentation, I actually want to talk a bit about the work that we are doing in UPM. So this paper is on Klebsiella pneumonia phage. We call it UPM 2146. All right, and what we did was actually, this is just the normal conventional method which I mentioned just now, where you isolate phages. I think this was from, I can't remember, pond water or drain water or something. All right, so we isolated phages against Klebsiella, then we did characterize it using the one-step growth curve, and we did the lytic efficiency to make sure. So this is an example where, without the phage, you can see that the Klebsiella pneumonia keeps on growing, but once you put the phage in, it's really, so this is a very good lytic phage. We did whole genome sequencing to make sure that it's lytic, does not have any toxin gene. All right, and then we did like phage stability test to make sure that that is to see where it's stable. So we did this for pH. This is the graph here shown is for pH. We also did it for temperature, and we can see that it's quite stable. And then we did TEM to see what kind of phage is it, the morphology of phage. So this is a very typical type of research for phage. All right, these are the kind of research that you can do. But the next part, I actually want to talk about phage-derived proteins because this is actually primarily my field of research. I'm actually a molecular biologist, so I do a lot of genetic engineering work. So I work more on the proteins compared to the phage itself. All right, so the title of this project was Lactococcus lactis, secreting phage lysins as potential antimicrobials against multi-drug resistant Staphylococcus aureus. So S. aureus is another bacteria which has gained a lot of resistance and is another bacteria of concern. All right, so talking about phage-derived proteins. So there are two types of phage lysins. One is called the virion-associated peptidoglycan hydrolase, and the other one is called the endolysin. All right, so these are phage-encoded lytic enzymes that can degrade the peptidoglycan or the cell wall of bacteria, and therefore they cause lysis in the bacteria. So these are the enzymes which are actually responsible for or giving the ability for the phage to actually lyse the bacteria. Okay, so the difference between VAPGH and endolysin is the VAPGH actually works during the infection and when the phage actually enters into the cell. So the lysis is sort of localized. It makes a hole for the phage to go into the bacteria cell. And then once the phage has actually replicated and it's time to be released, then the endolysin will sort of release into will break the cell wall so that the progenies can be released. All right, so endolysin works with another protein called the choline. The choline is a protein which makes a hole in the inner membrane of the bacteria so that the endolysin can break the outer cell wall. All right, so because if you in naturally, right, the phages is inside the cell during infection, and then it has to come out. So there's the membrane layer to go through, and then only you reach the cell wall. But what happens is that researchers have found that if you actually apply the endolysin from outside, there is no cell membrane, so you don't need the choline. You just need the endolysin, and the endolysin will break the cell wall, and you can use that as an antimicrobial as well. All right, the other difference is that the VAPGH does not have a cell wall binding domain, but the endolysin has a cell wall binding domain. All right. So the term that has been coined for this is actually enzymes. Enzymes act like antibiotics, so they are called antibiotics. All right. So this is what I was talking about. VAPGH is in step number 1 when the phage infects the bacteria, and then the endolysin is in step number 6 where it breaks the cell so that the progenies can be released. And then here you can see that the endolysins have they can have 2 catalytic domains or 1 catalytic domain or more, but they usually have a cell wall binding domain, while VAPGH does not. Reason because it's the cell wall binding domain is usually related to the specificity of the endolysin to see where it can, which kind of host range it can actually lies. So in terms of VAPGH, they usually do not need a CBD because the target is determined by the tail of the phage. Okay. So Lactococcus lactis is my main field of research. I'm still working on it now. So this is a gram-positive, generally regarded as safe bacteria, commonly used in food products. It's a model organism when it comes to genetic engineering for lactic acid bacteria. And it's highly researched as a microbial cell factory and a delivery system. So the idea here is that you can, because this Lactococcus lactis is it can survive the gut, you can actually use it as a delivery system for either vaccines or therapeutics through oral delivery. So you produce, you make or you engineer the LAB or the Lactococcus to produce whatever you want to produce, and then you can deliver it through oral delivery. It will bring it through the stomach, survive the stomach environment, and then go to your gut, and it can do whatever it's supposed to do there. Okay, and it's an alternative to E. coli host expression. We have found that there are certain proteins that sometimes are not expressed well in E. coli because of inclusion bodies or certain problems, but it can be expressed in Lactococcus lactis. So we use them both interchangeably, E. coli as well as L. lactis. Okay, so the question I often get is, why do I want to produce Staphylococcus phage lysins in LAB? So what's the relationship between Staph aureus and lactic acid bacteria? So although Staph aureus is usually known to be connected to your skin, but it actually also causes gastroenteritis, and so there's a there's a desire to actually eliminate S. aureus from food source. And lactic acid bacteria is also used as a starter culture in many food sources. So the idea is that, if you have lactic acid bacteria, which can inhibit pathogens, then that's desirable. Also, there are many cosmetics nowadays that contain probiotics. I think a lot of the girls here or the women here will attest for this, right? So yeah, so we have, imagine if you have a probiotic which are producing phage lysins, which can inhibit other bacteria, which causes things like pimples and acne on your faces. So this is the general idea lah. Okay, so the research approach is to express endolysin or VAPGH from the Staph aureus phage into Lactococcus lactis. So we have the gene, and then we fuse it with a signal peptide. so that the protein can be secreted from the cells, all right, and then we transform it into L-lactase. Okay, so this is just to show the expression of the VAPG in the endolysin. We have intracellular and extracellular fraction. The extracellular fraction is smaller than the intracellular because when it's secreted out, the signal peptide is being cleaved, so it's slightly smaller than the extracellular fraction. Sorry, slightly smaller than the intracellular fraction. All right, so, and then you can see here, this is for VAPGH. So we have all the different like buffers, different controls, but the one that we can see here is that the VAPGH, which are from the extracellular fraction, is have a halozone similar to the positive control, which is just chloroform, to show the lytic zone. All right, and then the same goes for endolysin. So the extracellular fraction is functional, but the intracellular fraction is not functional, probably because the signal peptide has not been cleaved, so it's not in the mature form. All right. So we also found out that this VAPGH and endolysin, they are dose dependent. When we increase the dosage, the zone of inhibition also increases, but the endolysin is more inhibitory compared to the VAPGH. And the endolysin is shown in the darker blue. And then we did the similar like kill assay, like a phage. You have the bacterial cells by itself, and then you have the bacterial cells which are treated with either endolysin or VAPGH. So the orange line is the one for endolysin, and the gray line is the one for VAPGH. So once again, we can see that the endolysin is much more lytic than the VAPGH. All right, so this is done using purified protein. We have the recombinant protein, we have expressed it in the lactococcus cells, then we remove it and we purify the protein so that we know for sure that the lytic capability is from the lysins, the phage lysins itself, and not from the bacteria or other stuff inside the host cell. Okay, so this is from purified. But the whole idea of using lactococcus is because we want to actually use it as a culture. So remember when I said that, you know, in phages, they have this capability of auto-dosing. When we use phage-derived proteins, we are actually, the good thing about phage-derived proteins is that they do not, so far, there's no reports that it will actually induce any resistance compared to phages. So phages usually will somehow or other, after some time, you also get resistance of the bacteria to the phages. But for proteins, lysins, so far, there has not been any reports on resistance. So that's good. But at the same time, when we use phage-derived proteins, we are losing the auto-dosing capability. So by putting it into Lactococcus lactis, we are hoping that the Lactococcus lactis, because it can grow and keep on producing the protein, you do not have to keep on adding the antimicrobial, the lysins, okay? So the idea is not to use the purified protein, but to use the cell culture itself, of the lactococcal cell culture. So what we did here was we used the cell-free extracts from the lactococcus. So the lactococcus secretes the lysins, all right? And then we take the lysine and then we culture it together with the Staph aureus and we see whether there's a reduction. So we saw that over 12 hours, it was a reduction up to 3.5 log reduction for endolysin. But this was the maximum that we can get. So we can see that it does not really go all the way down, all right? 3.5 log reduction, if we compare to previous studies, it's more or less there are some studies like here, which are more or less the same. Like for example here, LB plantaris secreting this endolysin, 2 log reduction in 24 hours. But there are also some which are very good. For example here, 30 minutes, but this is for purified protein. Okay, this one 4-log reduction in 30 minutes purified protein again. All right. And then we also tested it against different hosts of bacteria that we have in our lab. So the endolysin was more able to lyse multiple Staphylococcus strains. These are all clinical samples that we got from Hospital Sedang. And it can also lyse Staphylococcus epidermidis, but not the other non-related strains. While the VA-PGH could not lyse all these, it could only lyse the host cell itself. So this is another advantage of endolysins. While the phages are usually very, very specific, they can only lyse maybe 1 or 2 strains of bacteria. For example, the research that I shared earlier, right, on the Klebsiella phage, yeah, this one. This particular phage that we had, I think out of 20 over strains that we test, it could only lyse maybe 2 or 3 different Klebsiella pneumonia strains. So even other Klebsiella strains couldn't lyse. Endolysins, on the other hand, they are much more wider host range. They can usually lyse up to the genus level. So which is why here you can see Staph aureus and also Staphylococcus epidermidis. So but then usually it won't lyse across the genus. So like Streptococcus and definitely gram-positive and gram-negative, very hard to find endolysins we can lyse both. So in that sense, it's the target is quite ideal because you want it to lyse at least all the bacteria in the same strains or the same genus, but not across it. Okay. All right, so we successfully managed to produce Lactococcus lactis, which can secrete phage antibiotics, and they can act as antimicrobials. However, there's also challenges because we want to improve the lytic capability, and we also think that the production, I think one of the reasons why when we use the culture, the cell-free extract, and it does not lyse as well, is probably due to the production of proteins as well. So this is a problem that we usually face in L. lactis compared to E. coli. The expression is not as high, and the secretion also needs a lot of work. So there are very few, compared to E. coli, there's a lot of genetic engineering tools that we can work with. We can use all sorts of signal peptides, we can use all sorts of expression vectors, and there's so many things to play with. But with Lactococcus, if we want to change the vectors or the promoters, we actually have to do it ourselves. So to engineer the promoters and stuff like that ourselves, so it's a bit challenging. Yes, so we are also working on that. So this is a paper if you are interested to look more into the results that I showed just now. Sorry, I think okay, these are the acknowledgement for the papers just now. These are the people who were involved. Charu is the student who did most of the work there. And then I work closely with Prof. Raha and Prof. Khatijah, and also Dr. Erin. So ongoing research that we are doing currently in the lab, we are isolating and characterization of phages and endolysis against aquaculture pathogens. So from Staph aureus, we have moved to aquaculture pathogens, including Streptococcus, Agalactiae, and as well as the Pseudomonas. And then we are using the same system, Lactococcus system, to deliver endolysins, but against other pathogens. And then I also have students who are doing mutation studies on the endolysins to improve things like the binding affinity and the host range, and also to increase the lytic activity. So we are trying many types of mutants, but this is also another very challenging project. It's very hard to, theoretically we think that it will work well, but after doing some mutations, sometimes it doesn't work as we expect. So but yeah, this is part of research, right? We keep on doing it again and again. And we are also trying to engineer gram-negative endolysins to penetrate the outer membrane. So one thing about endolysins, they work primarily for gram-positive bacteria at the moment because we are applying it from outside, and gram-positive bacteria do not have the outer membrane, so it works well. But for gram-negative bacteria, you have to think about the outer membrane as well, So we need to engineer things like peptides, which are able to destabilize the outer membrane. So this is something that we are also working on in my lab in UPN. All right, so before I end, this is the end of my what I would like to, I want to do a bit of advertisement. We are having a lactic acid bacteria conference in Kuching in October. So anyone here who is interested, this is a face-to-face conference. So if you are sick and tired of virtual conferences, or you can come and register for this conference. All right. So thank you very much. I can take questions now.
Okay. Thank you, Dr. Adeline Song, for your excellent and informative talk. Thank you for starting your presentation with a little bit history of the discovery of antibiotics and also MMR, and also the pros and cons of the Phage detection methods. And most important, your sharing on your works, very interesting, with the VAPGH and also endolysin. And also we know that Lactococcus lactis will be a very good factory for the production of the antibiotics. So now we will start our Q&A session. Dr. Adeline, may you please stop the share?
Okay.
Yeah. Now we will start the Q&A session. So there's a lot of questions pop up from the chat box. Yeah, so we will start with the first one. So one of the participants asked that, is it advisable to use bacteriophage as an alternative of antibiotics in poultry industry? So how effective it would be compared with the use of antibiotics?
Okay, I think it's pretty much similar to all the other industries, not just poultry. Personally, I think I had one student which I co-supervised who actually tried using phages to eliminate bacteria from, but it wasn't in live poultry, it was in chicken skin that she got from market and all. So yes, it's at the moment, right, I think, and it's the same as all the other industries, because phage therapy is still in the research stage, right? So it's not very established, and there are many many unknowns, all right, and also many contradicting or reports. So some say it works well, some say not so much, because maybe like we mentioned just now, right, there are certain phages who are very specific against only certain bacteria strains. So sometimes, you have to identify very specifically what type of infection is it first before you can actually identify phage, the phage that can actually work. So it's sort of like a customized kind of a therapy. So if you want to do a one step kind of approach where one cocktail can lyse almost everything, it has to be a cocktail of phages which are slightly broader. Okay, so to be honest, at this moment, I think we are not at the stage where phage therapy can overtake antibiotics. Antibiotics are very, very effective. Okay, but it's just that we are running out of options with antibiotics. So when it becomes ineffective, then we need alternatives. And also, previously I shared all the other strategies, right, that people are using as an alternative to antibiotics. It's not that I wouldn't also say that phage therapy is superior compared to all the other strategies, but we really need all options possible. All right, so there is a potential in phage therapy. We have we mentioned just now, it's so abundant. Whatever we know about phages is probably just like 0.2% only. There's so much more phages to discover, and we might find new findings. So I think the idea here is that we need to have many different types of strategy when we combat AMR. So it's not just about finding new alternatives, we also have this initiative called the One Health Initiative, which looks into all aspects of AMR, like surveillance, where all your antimicrobial bacteria is coming from, going where, that kind of thing. Talking about different industries coming together to combat AMR, having laws and to tell farmers that no, you shouldn't use antibiotics or you cannot use antibiotics. So we need all sorts of strategies to actually combat AMR. It's pretty much like the COVID situation, right? We are just vaccines alone is not going to solve it. It's only a certain percentage effective. You need things like SOP, you need things like social distancing. So you have to come at it at all different angles. Yeah.
Okay, thank you for answering, Dr. Adeline. So now another question. Since phage therapy is a good option and alternative to antibiotics, would farmers nowadays would choose this therapy as they are looking for cheaper and also accessible options?
Yeah.
Phage therapy would be a good choice for them.
Yes, I think so. I think farmers, they are very whatever works, right, and they're allowed to use it, they will definitely, right? Because if we just tell them don't use antibiotics, but there's no option given to them, it's very hard for them. And also it's got to do with food security, because when you ask them don't use antibiotics, then they have to take into consideration that, for example, we have organic food, right, in our supermarkets and everything, but organic food is always so much more expensive than the normal ways that people the normal chickens or whatever. And the reason why is because they have to take into account that, if they are not using any growth promoting factors, they're not using any antibiotics, the chickens might easily be diseased, fall sick and everything. So they have to take into account those laws. So their price is always higher, right? So so I think if farmers are given alternatives, they will definitely use it. But it has to be cheap, it has to be effective, and also easy to use. So a lot of farmers nowadays are actually using probiotics, especially aquaculture farmers, to replace like antibiotics, right? But the other issue is that, it's also got to do with educating. So for example, because currently I have a project on phage therapy and also lactic acid bacteria in aquaculture industry. And so I'm not a fish person, but then my collaborator is he deals a lot with these farmers. So he was telling that a lot of these farmers, because they don't know what probiotic is, they just know that, you put it into their farms, it can help their fish. So they are actually, sometimes they use both antibiotics and probiotics together. So antibiotics are actually killing their probiotics, but because they don't know that the probiotics is bacteria, right? They don't know how it works. So things like that we actually need to educate the farmers as well. But yes, I think that if it works, then they are definitely on board.
Yeah, understand. Thank you, Dr. Lin, for your answering. And then we have another question. Someone is very interested in your research. They want to know that how well your bacteria, like the lactococcus, will compete with other good bacteria. If the person consumes the capsule containing your engineered lactic acid bacteria, will there be any competition with their existing probiotics?
Okay, good question. So the strains we usually work with is Lactococcus lactis. And although we call Lactococcus lactis in papers, right, we call it like probiotics because they have the beneficial properties. But if you talk to people who are in the human nutrition kind of people, they will not call Lactococcus lactis a probiotic because it doesn't fulfill the definition of it what's the word for it, colonizing your gut. So compared to lactobacillus, it actually colonizes your gut. Lactococcus lactis is able to survive the gut, but it actually passes through. And this is the reason, one of the reasons why we use this, because a lot of our work is got to do with genetic engineering. So we, at this point, we don't want it to be, permanently in your body. We want it to go in, do what it's supposed to do, and then come out. Yeah, so it's more for safety purposes, also for us to monitor lah. So yeah, so at the moment it's when we use Lactococcus lactis, then it's non-colonizing. So it will actually go in and come out. If you use Lactobacillus, then it will colonize. So there's also, of course, concerns about GMO. So we also have looked at certain ways to make it non-GMO. For example, we had a project previously where we expressed the proteins of interest on let's say a different E. coli cell, right? We just take the protein and then we use a display system in Lactobacillus or Lactococcus, and we just display it on the outside of the cell. So the cell just carries the protein in and is decorated on the outside of the cell, but because it's not engineered, so there's no GMO involved lah, in this sense. So it's more for a different strategy in case people are very adverse against GMO kind of thing. Yeah.
Okay, thank you, Dr. Dalim. So there's another question. So given the reduced rate of expression, so how many liters of culture media do you use usually to produce the desired amount of proteins or cell-free extraction?
Okay. So depending on what kind of proteins are being expressed. So like, for example, while I was doing my PhD, right, I was expressing like plant proteins in Lactococcus, so like to get super the amount of proteins that I needed, I had to go up to maybe 3 liters of culture to purify. All right, but I think for this, the one that I showed just now, the Phage one, if I'm not mistaken, it was I can't really remember. Probably in the paper you can find it, but it wasn't a lot because we just took the cell-free extract only. So there was no purification or anything involved. So we just took the cell-free extract, maybe in 100 mils or something like that, or yeah, around that. Yeah, so not so much lah. So that's possibly also why the rates didn't reduce as much as we would have expected it to be. But when we use purified protein, then the concentration is very high. Then that was like, I think from 1 liter, we concentrate it further into maybe a few mils, then we get more protein, very concentrated protein. Then you can get better lytic activity as well.
Okay, thank you. So we have a question from another participant. She commented there's a great sharing from Dr. Adeline. And so regarding your research, did you also test it on human cells or cell line infection?
No, we haven't tested our Phage project on any human cells or cell line. We have the Klebsiella one. We use in vivo model of zebrafish to test the toxicity. And then all the other projects that I'm dealing with, we will actually test it on aquaculture, means I will be testing it on tilapia fish lah. And yeah, so that's also the reason why I actually chose diseases that has got to do with animals instead of humans. I'm moving towards that because it's very hard to get funding and mice work and all cost a lot of money to do our animal studies there. And usually when we have animal studies in mice, it's very hard to replicate it. Once we have done it, then if the extended duration is too high or what, it's very hard to get enough fundings to actually redo everything until we get good statistical data. So we want to test the Lactococcus system that is delivering the Phage. We are gonna do it in something cheap like tilapia fish first, so that we can do many, and then later on once we have the system in place, then we will come back and yeah.
Okay, thank you. And we have also a question from our Dean of Faculty of Life Science, Ames. So he want to know that what method you use to genetically engineer lactic acid bacteria. So how long it takes?
Okay, so we do the conventional method where we have a plasmid, the vector, It's a commercial vector. You can actually purchase it. So there's in Lactococcus lactis, there's this nice system. It's the plasmid is inducible by niacin. Niacin is also a food grade vector. So you can just express whatever gene you want into the, clone it into the vector, transform it just like in E. coli. The only issue is that it's harder. All my students who have worked with lacto and E. coli, it's harder to do the cloning in lacto compared to E. coli because of the one thing is that the cells grows a bit slower than E. coli, so we have less proteins, less of everything. So for example, the doubling time of E. coli is 20 minutes, the doubling time of lacto is about 40 minutes. So when we grow, sometimes when you do plasmid in E. coli, you probably need only 1 mil. In lacto, you probably need 5 mils, that kind of thing. So yeah. So but then the method is pretty similar as what we do in E. coli using a plasmid expression system and all those.
Thank you.
Okay, thank you for your answering, Dr. Adeline. I think it's the last question from the participants due to time limitations. So you want to know that how do you motivate yourself through your research and your careers? Yeah.
How do I motivate myself? Yeah, okay, research is really hard. I have to tell everyone, right? Because that's why it's called research. You keep on searching, right? So a lot of times things just, especially research, it just never goes according to plan. But I think what students have to remember is to always keep, when you do your research, you do it properly. Make sure your methods and everything are valid. Make sure that your results are consistent. There are reliable results, can be reproduced. Okay, and if things doesn't work out the way it's supposed to work out, that's fine. Sometimes you get negative data. That's also part of research. I think the issue now is that people are so pressured to publish to get good results and everything that we tend to make our results biased, towards what we want it to be, and I think that's really hurting our research as well. So I hopefully that everyone can maintain your spirit to be to do research with integrity, right? And I think in terms of motivation, you need to have a goal, and it's okay if you sort of reach your goal later, but you have to keep going at it so that you don't get lost. Remember, you can change your methods on how you reach your goal, but you should reach your goal right? So you have to be flexible and you have to persevere. So I always tell my students, if you want to do your PhD, it's not the smartest people who will make it. It's the person who perseveres the most, right? All right.
Okay, thank you, Dr. Dalin, for your kind word. So we would like to end these sessions now, the Q&A sessions. So thank you again, Dr. Dalin Song, for your wonderful talk and for your sharing your thought with us. So I'm sure that everyone have a fantastic session together, and it's a very fruitful talk and interactive session. So thank you to our chairperson, Vinodini Kunalan, and the organizing committee for the wonderful job. And also special thanks to Prof. Ravi for encouraging Year 3 Biotech and Bioinfo students to organize the webinar. So now we have our chairperson, Vinodini Kunalan, who would like to deliver a thankful speech. So Vinodini, please.
Thank you, Ms. Chai. On behalf of APSA and the organizing committee, first I would like to thank Dr. Adeline for an enlightening and informative talk on phage therapy. We are very grateful for the time and effort you took to share your thoughts and experience with us. We really appreciate it. And thank you very much to our Senior Professor Manikam Ravichandran, Dean of our Faculty of Applied Sciences, for attending and supporting our webinar. I believe that this webinar helped you to get some insights and learn something new. Once again, thank you very much. And over to you, Ms. Chayuchi.
Okay, thank you, Vino. So as a moderator of today's webinar, I really enjoy it and hope to invite Dr. Adeline Song to join us again. So now we will be having a photo session. So everyone, please turn on your camera and have a picture together. Hi everyone, please turn on your camera. We have a photo session now. Is everyone turn on your camera? Okay, now taking picture now. Okay, thank you. All right, ladies and gentlemen, before ending this session, we would like to hear feedback from all participants by scanning the the code on the screen right now. So we promise that the evaluation survey will not take you more than 1 minute, and your feedback will be greatly appreciated. You can either scan the QR code or go to the link below, and the link will be provided in the chat box as well. After the completion of the survey, you will receive an instant participant certificate in a short while. So, and I will keep this slide running for 2 more minutes for you to scan the QR code. And we will looking forward to having you join us again in the next session. And thank you again, everyone, and wish you good health and safety.
Thank you, everyone. Bye.
Thank you, Adeline.
Thank you,
Dr. Adeline. Okay. I hope everyone has the chance to scan the QR code, and I'm going to end the meeting. See you again in our next webinar. Stay safe and have a wonderful day. Bye-bye.